Welcome back, biology. Today we're going to talk about meiosis. We're going to really simplify it. All I really want you to know at this point is the end results, why a cell would undergo meiosis, and what's happening in each of the phases of meiosis. So, what is meiosis? It's a form of cell division. We've already talked about mitosis, so similar concept. Cells are making new cells. But the important part of meiosis is that it is going to have the number of chromosomes. So this is important for reproduction. You have 46 chromosomes in your cells. If your cells were fertilized by another 46, your offspring would have 92. It wouldn't even be human, really. So if that one continued to be fertilized, 92 and 92, 184. So generation after generation, you just get too many chromosomes for your cells to know what to do with. So we have to cut the number in half. So you put two halves together during fertilization and you'll have a whole. So that's the really important thing. So this is used when making gametes or spores. And I'll put a little video animation in the show notes below so you can click on that URL address and check it out. PMAT, same phases as we use with mitosis, but we're going to do it twice. So we have prophase, metaphase, anaphase, telophase. Repeat. So first of all, you have prophase. Notice you have chromosomes that are paired up. The sister chromatids, those identical chromosomes, are paired up at their centromere, but they've come to find two others just like them. So homologous chromosomes have synapsed. They formed a tetrad. And so you'll see here pieces of those homologous chromosomes get tangled up. And when they get tangled up, eventually they get pulled apart and pieces break off and get switched onto the wrong chromosome. And so that's going to create some genetic variation. This chromosome will no longer say what it used to. It'll say something different. So this is what that looks like. Remember, sister chromatids are identical chromosomes. So during interphase, this light purple one made an exact copy. This dark purple one also made an exact copy. So the relationship between this sister chromatid here and this one here, they're identical, so they're called sisters. Over here, these carry the same information as far as what traits. So maybe this is your hair color, your eye color, and the number of fingers you're going to have. This one also carries information for hair color, eye color, and number of fingers, but it might carry different information. So this might say blonde hair, this says brown hair. This says blue eyes, this says brown eyes. This says have 5 fingers, this one says have 6. So the traits are the same, but how they're coded for is different. So they're homologs. They're similar, they're not identical. So crossing over can only occur between homologs. Pieces of the chromosome gets broken off, and you can see this light purple got attached here. So now it says brown hair and blue eyes instead of brown hair and brown eyes. Okay, so they've switched spots, and that's called crossing over. So that's increasing genetic variation. A little bit more with those homologs, because I know students get confused. Just know that sisters are identical. Not only do these have the same color, but they have the exact same code. So you can see a capital A here, a little b here. These are similar. They're the same length. They have the same centromere position. They're carrying information for the same traits. The information isn't the same. This one has a little a instead of a big, has a big B instead of a little. So you see they're similar but not identical. So the two purples are sisters. The purple to the blue is a homolog. And this is showing crossover where the B's got switched. So now it's no longer little a, big B, it's little a, little b. So that makes a new genetic code. Okay, metaphase. Homologs line up at the equator, right here, homologs. Groups of tetrads, see 1, 2, 3, 4. Tetra is 4. So this set is called a tetrad. It is 2 homologs, or 2 chromosomes. So they're going to line up at the equator just like they did in mitosis. They're connected to chromosomes. The only different than mitosis is that mitosis was a single line and this is a double line. And again, I'll show you a little animation link that you can use later. So anaphase 1 chromosomes are being split. Different than mitosis. In mitosis, you had individual chromatids splitting. This time you have entire chromosomes. So notice the homologs were lined up across from each other. So mom's and dad's chromosomes are separating. At this point, we are having the chromosomes. So this is called a reduction division. You've gone from a 2n number to n. Telophase, going to split the two chromosome or the two cells in half, create two new daughter cells. At this point, they have half the number they originally did. We started with 4, we now have 2. The cytoplasm divides, we have 2 new cells, and they're going to go through the phases again. This time, we're not going to precede it with interphase. There's not going to be any replication. So we still have 2. These 2 cells are going to go through PMAT all over again. So spindle fibers will reattach to chromosomes. Chromosomes will line up at the equator. Notice this time we have single file lines instead of double. So we're dealing with sister chromatids. Notice the sister chromatids in Anaphase 2 are separating, looks similar to mitosis, only I have half the chromosomes that I would have had. So a big key when identifying cells verbally, if it talks about homologs, you know you're referring to the first set of phases. If it talks about sisters, you're referring to the second set of phases. So the sister chromatids are now going to be split. They're no longer linked to their partner, so they're no longer called sisters. We'll just refer to them as chromosomes from now on. So when these cells split, you get 4 totally new cells, each with a haploid number. So we started with 4, we ended with 2. Nothing has changed since the end of telophase 1. New nuclear membrane will reappear. Spindle fibers disappears. Chromosomes will uncoil again and form chromatin. And you once again have 4 haploid daughter cells, each with some genetic variation due to crossover, due to the fact that these will line up differently during metaphase and may separate in all kinds of different combinations where you get one of mom's, one of dad's, one of mom's, one of mom's, one of dad's, one of dad's, different one of mom's, different one of dad's. So there's all combinations possible. And again, I'll put a link for you to watch an animation of that. So 2 important things are happening during meiosis. The biggest thing, I think, is that it's causing the chromosome number to cut in half. So for us, we have 46 chromosomes, and you cut it in half, and they'll have 23. Any organism has an N number. So that N is a variable, right? So every organism has a different number, every species. All humans have 23, so 2N. Our diploid number is 46, 2 times 23. But an organism that maybe has an N number of 14, their 2N number is 28. So their body cells would have 28, but their sex cells would have 14. So it cuts the chromosome number in half, so when that cell is fertilized, it has a full set once again. The other thing that's happening is we're getting some genetic variation due to independent assortment of chromosomes and crossing over of homologous chromosomes where pieces get switched. And so they create 2 different variations of the same species or the same organism. So comparing it to mitosis and meiosis, just a summary here. Mitosis creates diploid cells from diploid cells. Meiosis creates haploid cells from diploid. Mitosis occurs in somatic cells. Those are body cells. It's every other cell other than a gamete, a sperm or an egg, a spore. Okay, it's all of the body cells. Whereas meiosis occurs in germ cells. Those are the cells that create reproductive cells. In the cell division of mitosis, we end up with a 2 in number, and that is identical to the parent cell. Whereas in meiosis, we end up with an N number, which is not identical to the parent cell. It has a number of different variations possible and a number of different combinations of alleles on the ends or wherever of the chromosome that might have swapped spots. So really you have a mix of mom's and dad's genes. You don't have all of one, you don't have all of the other, you don't have all of both. You have some from mom and some from dad. And so that is meiosis in a nutshell. So I hope that was helpful. Study your phases, be able to recognize them by picture recognize them by description, and know the difference between mitosis and meiosis. So I hope that was helpful. Check out the links below. Animations are useful. Thanks for watching.